So welcome to today's webinar, which is being hosted by the Sprint project. So Sprint is a Horizon Europe funded project, and we are researching the impacts of pesticides and plant protection products on various metrics, so including environment, health, animals, and beyond. So today we are focusing on the sediment side of our research. So we have Vera Silva here, who works on the Sprint project and is based at Wageningen University and Research. She's going to share insights about one of our recent Sprint peer-reviewed articles. So the article is entitled "Pesticide Residues in European Sediments, a Significant Concern for Aquatic Systems." Vera is going to then be joined by Aurea Hernández, who is based at the University of Bern. So Aurea specialises in the analysis of organic contaminants in natural systems, including sediments, and also their effect on microbial communities. So just to remind everyone before we get started, please use the separate Q&A function for questions and use the chat for discussion. We'd love to hear from you as much as possible. We want this to be an engaging webinar. And it's also worth noting that the webinar is being recorded. So if you're supposed to be somewhere else, keep your camera off. So excellent. Without further ado, I think we'll get started. So I'm very pleased to introduce Vera.
Thank you, Charlotte. I will just... Hi, good afternoon, everyone. Thank you for joining us. I'm just sharing my presentation. I hope you can see it now, full mode.
Yes, almost.
Yeah?
Yes, now, perfect.
Okay, super, thank you. Okay, yes, so thank you also for the introduction. My name is Vera Silva. I'm a researcher at Wageningen University, and I'm in the coordination team of the Sprint project. Yes, so a quick catch up for those that are not familiar with the project. This is an Horizon 2020 project that started in 2020, and it runs until the end of summer of upcoming year, 2025. And this is a project on the umbrella of integrated health approaches and alternatives to pesticide use. So we are rather advanced in project development, and also why this series comes now on our publications. And I put here in this slide some of the highlights of this project that goes through the fact that we have an holistic and One Health assessment approach, that we have unique data sets and approaches in the lab. Focus here on the fact that we test mixtures of pesticide residues. Also on the fact that we have a toolbox, a global health risk toolbox, as a key output of the project, and also that we not only look into the impacts of pesticides, but also work towards the transition towards more sustainable pesticide use and food production. And ending the slide with our train, the all on board on this effort, and all the actors in the food chain to help here. So I'm starting with a very short introduction, more direct already with the sediments, so that that is the focus of today's webinar. So I'm not going in deep now on the advantages and disadvantages, or like pros and cons associated with pesticides, but stating that, well, we do know, and there are several evidence that show that pesticide residues can be transported from the fields where they are applied during or after the application through different processes. Of course, for water bodies, the most relevant ones are the surface runoff, if it is surface water bodies, or leaching for groundwater. We do know this is rather complex. This fate of pesticides is rather complex and governed by different factors. So in regards to transport to water bodies, the soil type, landscape characteristics, pesticide properties, and management practices are highly relevant to explain and understand the findings in the water bodies. And I think this highlights also one of the strengths of the project, that that is this holistic assessment, that indeed we need to put a pesticide as a holistic challenge and look at it integrated across matrices and compounds. And that's what we did in 2021, right at COVID times. We did a comprehensive sampling campaign. And we collected different type of samples all around the same time at the middle of the growing season, where the diversity of pesticides applied was expected to be the highest. So we were looking for a worst case scenario. And we also had this, so we ran this monitoring in the different case study sites, also using same monitoring program, so SOPs to harmonize as much as possible and make available comparisons. So here in this slide, you have on the left a representation of our sprint case study sites. So we have 11 in total, 10 in Europe, 1 in Argentina. Argentina here as importer of feed to Europe. And altogether, this covered the main crop types in Europe. And in each one of these case study sites, we have both conventional and organic production. And in each one of these case study sites, we did a rather comprehensive sampling campaign, which you can see the icons in the right. So we sampled samples from human participants, so farmers, neighbors, consumers, and these samples went from urine, blood, feces. We asked them to wear wristbands, also asked them to provide food, a portion of the food that they ate for analysis of pesticides. We did also a comprehensive campaign at the environmental level, water, soil, sediments, indoor dust, and so on. And we also collected samples from livestock, so that altogether we could understand, characterize, and explore distribution and health status of these case study sites. Here, some pictures of the field campaign across the different case study sites. And here, the number of samples that we have collected. So I highlighted here the sediments. So it was a kind of exploratory assessment. And by the sediments, I'm talking about the waterbed sediments. So in the water bodies, we collected those sediments. And we managed to collect 38 samples across the European case study sites. And that is the focus of this paper that I am presenting today. So earlier this year, we published this publication, Pesticide Residues in European Sediments in the Environmental Research. And I'm going to guide you here on the rationale, the methods, and the main findings of it. So, rationale. Why is relevant to account also for waterbed sediments? Well, because it is known that they can act as sinks, but also a source of contaminants. The type of compounds in these sediments, it's highly dynamic with the characteristics, and depends, indeed, these characteristics decide if they go into water, they salt phase and present in water, or if instead they have higher affinity for organic matter and they can absorb and accumulate in the sediments. What is known is that indeed there is this dynamic between the salt and settled phase, and that the pollution of waterbeds and water bodies in general may have negative adverse effects on the functions of the aquatic ecosystems. So here in Sprint, we accounted for waterbed sediments to help address pesticides fate, pesticide persistence, and also infer on potential consequences to the ecosystems. These ultimately can help support decision-making and also more sustainable land management and protection of these aquatic ecosystems. I'm presenting today on the results of pesticides in sediments, but this comes in a package, let's say. So I already published the results of pesticides in water, and we are also working on the publication of the pesticide analysis in fish and also on the macroinvertebrates diversity. So those all together to help characterizing the aquatic environment. So the sediment samples, well, we focus on small, they came from small water bodies. So that is a big difference from most of the data that exists in literature. So here we have small water bodies that include small reservoirs, lakes, ponds, rivers. So as you can see, we cover both lentic and blotic systems. And the criteria was that they would be small, but also connected or in the close vicinity of agricultural fields. We found 38 locations where we managed to collect samples across 8 of the 11 case study sites that we have in Sprint. Each one of these 38 sediment samples are in fact a composite sample. So there is, each one of them represent a water body, and we collected 3 subsamples along that water body, which we combine in proportional, in the same amount to get this composite sediment sample. We focus also on the top layer of these sediments, so from 0 to 10 centimeters deep. So indeed, the ones that are also more prone for being remobilized, resuspended, with operations management and the current of the water body. In each one of these sediment samples, we analyze 196 pesticide residues. And by pesticide residues, I mean active substances, but also metabolites. And this list of compounds was put together based on our previous knowledge of applications in these regions, but also in terms of what is expected in the crops that we sampled, application records from the farmers, but also we did a complete full screen of some samples to make sure that we were not covering relevant compounds. So on top of the pesticide analysis, the sediments were also analyzed for basic characteristics, pH, organic matter, and texture. And on top of these, we retrieve some information on these pesticides that we were targeting, namely approval status, if they are approved or not in the European market, the degradation rate, so how persistent they are in this water sediment nexus, and also if we knew these compounds to be applied or not in this region. And this we have the application records from the farmers of the connected agricultural fields. We did rather exploratory and basic descriptives on the data, and we analyzed it mostly at 2 levels, sample and matrix level, and I put here in bullet points the endpoints that you can find in the publication, but I'm also highlighting them in the slides to come. So here, first finding on the number of pesticide residues in the samples. So here in this graph, you can see 38 columns for the 38 sediment samples. And on top of the graph, you can see the codes for the countries where these samples originate from. And the colors in the bars indicate the persistence pesticide residues found there. So what do we see overall? Well, first thing, we see 2 big bars in Czech Republic. So 2 out of the 3 samples collected there actually had more than 45 pesticide residues. We also see that there is only 1 sample in Slovenia, sample 15, also highlighted here by a blue arrow, that it was the only sample that was pesticide-free. And I put pesticide-free between, not brackets, but as a quote, because, well, we did test a lot of pesticide residues, but it does not mean that others that we may have not tested are not there. And this, of course, applies to this sample and to all the others. We noticed also that the vast majority of the samples had like relatively simple mixtures. So 13 samples had 2 to 5 pesticide residues, 7 more samples had between 6 to 10, and then from 11 to 20. So those were the most common scenario in the sediment samples. When looking into the colors of in the columns, so the persistent, we do see also that actually it's rather common to have a mix of all these classes, and it's not obvious a predominance in terms of country or and keep in mind that a country here is also linked to a crop system. So there is not a clear dominance. There is indeed a mix of the different classes all over the European water bodies and sediments tested. We looked also into the levels of the pesticide residues, and here in this graph I show you the total pesticide concentration when we add up the different residues on top of each other, but you also see the 10 compounds that had the highest concentration. So you see glyphosate and its metabolite AMPA, we have DDE and DDD, so DDT metabolites, and we have 6 more, the fluodioxin, azoxystrobin. So these were these that are on top of the graph, these 10 were the ones that had the highest concentrations. And well, we also look into the complexity of the mixture, so not only on the levels, the total levels, but also how complex these mixtures were. And you can see here that, well, not as unexpected, the ones that had more residues, that had also the most complex mixtures. But we also saw that there were 2 samples, one from France and one from Portugal, both case study sites that are dominated areas from vineyard production. And we saw that these 2 samples also have a high diversity of compounds. So indeed here, the crop production and the application pattern may be an indication of what you may find out in the sediments in these regions. So more on the pesticide findings. Overall, as I mentioned before, we tested 196. We found back 99 of these pesticide residues. 72 of them are approved in the market, which leaves 27 of them that are no longer approved at the sampling time. So that is also something to reflect on. We found 3 compounds to be highly frequent in the sediments across all the samples: glyphosate, its metabolite AMPA, and DDE. Those were common in almost all the samples, which highlight on one hand the high use of glyphosate-based herbicides, and on the other hand the high persistence of DDTs still in the environment. From the other compounds, we found rather sporadically, actually 59 of them were detected in less than 10% of the samples, and then 31 between 10 and 25. Although these were the most common, they are not necessarily the ones that have the highest median concentrations. In fact, cyprodenyl and dicamba were the ones that had the highest concentrations, median concentrations in the samples. Once again, here there were compounds that were found at rather residual levels, but please don't get my message wrong. Residual does not mean risk-free. Okay, so I'm just making here a statement in terms of relative comparisons of the compounds. Total pesticide content, as I showed you in the last slide already, it was in Czech Republic, where it could reach up to 1,200 micrograms per kilogram. And the lowest total pesticide content was observed in Slovenia, Switzerland, Croatia, and Denmark, where you can see here that the highest level was 1 order of magnitude lower than those measured in Czech Republic. We found also that despite all the variability in terms of composition and levels, that there was a strong and positive association between the pesticide levels and organic matter content of these sediments. We found also that mixture composition was very specific of the sample, but there were like some roots that were common. So there are some elements, some combinations that keep appearing across the samples. And that is what these graphs try to do. So we look into roots of these mixtures, so the core element of these mixtures, and we found that there are a combination of glyphosate and AMPA that were present in 23 out of the 38 samples. So that's what you can see in the right. And then you see the other combinations of two compounds, how frequently they were. So AMPA and DDE appear in 17, glyphosate DDE on 14, and so on. And once we increase, of course, the root, increase the number of compounds in this root from 2 to 3, and we include then on the bottom right, you can see a root of glyphosate, AMPA plus DDE, so the 3 more frequent ones. This combination was present in 14 out of the 38 sediment samples collected across Europe. So this indeed highlights some common composition in the mixtures that adds up to all the other practices. So, in terms of discussion, these findings, these analytical findings, corroborate off-site contamination by pesticides, namely in water bodies, corroborate also that sediments can serve as sinks of pesticide residues. We found I put a box in the right that highlight how much of a sink they are in terms of diversity of compounds found, the amount of sample of pesticide residues that you can have per sample, and also the level. So yeah, almost all the samples, actually only one sample was pesticide free. All the others had pesticide residues in a mix of approved, not approved substances and metabolites. We also found that the concentrations of pesticides in the sediments are most likely a result of how intensive and how diverse is the agricultural practices in that region, but also on the sediment characteristics that can affect severely the persistence of the compounds in the sediments. In terms of glyphosate and AMPA, so the ones that were the most frequent compounds, they were also rather dominant in terms of highest concentrations. And these, again, link to the widespread use of glyphosate-based herbicides, which at the same time hampers further analysis on the source of the contamination. And also the fact that, there are also all these our case study sites were mostly agricultural areas, but there are also potential input from urban pesticide use into these water bodies. On the pesticide-free sample, the only one, so that was found in Slovenia. So, and I'm putting here is an example of the type of rationale that we can have around these findings. So we look into the characteristics of the sample and where it was collected, and actually it was the one at the greatest distance from the agricultural fields. So that can be part of the reason why it was pesticide-free. But we would need to have more information on the soil types of all this region, or all this watershed, I mean, and also on the connectivity of these agricultural fields with the water body that has been sampled. This is a rather relevant factors to indeed understand the findings in the sediments. Other factors, of course, could have been raised, like the proportion of conventional and organic in the area, or the fact that the type of pesticides not being used could be raised, but because luckily we did an integrative assessment and this holistic assessment, we have findings also from the water, and in the water bodies there were pesticides there, so it was not that it was not connected at all, or there was not relevance in terms of pesticide use. So this comparison between water and sediments is also rather interesting. Last but not least, we had comprehensive, sure. We look into 196 pesticide residues, but it was still targeted. So it is possible that this sample, like all the other samples, may present other not analyzed pesticide residues. And this is my last slide about the implications of this study and this type of studies. Well, we see that there are compounds that are being applied nowadays, that are approved in the market and that are being applied nowadays, that can accumulate in sediments. So there are compounds of moderate to high persistence, like AMPA and azoxystrobin, for instance, that accumulate in sediments over time and may lead to concerningly high concentration. So, of course, the logical next step is assess the site-specific risk assessment and understand the risk of these levels. I focus here on sediment as a sink of contaminants, but we know that sediments are also a source. So that both anthropogenic activities and natural events can lead to resuspension, of the contaminants into the water body. And that is also relevant to account when you indeed want to protect and manage these water bodies. In situ, and ex situ impact, so in the water body, but also if these sediments are used elsewhere, for instance, in the case of the possible of dredged sediments, this needs to be accounted, this contamination, this complexity, these levels, and should be further explored. And last point, that all of this comes together and that we are targeting a higher protection and conservation of aquatic system, lower pollution all over, not only in water, but soil, air, water in the European vision. And this type of information is needed for informed decision-making and also to assess the impact of measures that are being taken. I think this is it from my side. Thank you very much.
Fantastic. Thank you so much, Vera. It's been great to refresh myself on this particular paper. And I think, as you say, there's a lot more research to be done, but the findings here are really important. So without further ado, I'm pleased to introduce Aurea, who is going to present some of her research around sediments and pesticides.
Yeah, thank you. I will share my screen. Okay, so hello everybody, and Charlotte, thank you for the invitation. My talk will be about the 60-year history records and fate of persistent or plant protection products in aquatic systems. From now on, I will call it PPPs and also pesticides to interchange. I will have to say that after listening to Vera's talk, I think my talk will challenge a bit Vera's talk. So I'm very looking forward for the discussion. So let's see if it works. Okay, perfect. So just to introduce a bit the Institute of Geography at the University of Bern, we have a cluster of environmental pollution, which consists in paleolimnology that we look more at micropollutants, time series, eutrophication, and anoxia, but also we have a group that look at more of the soils, metal, microplastics, and we also link it to the human geography. Within the context of this, in the University of Bern, we are interested to understand the effect of different environmental factors on food systems, from, for example, soils and sediments to human health. And this is where my research enters. And if I want to start and go back to the PPPs, and Vera already introduced very nicely what are PPPs and how these distribute in the environment. And the truth is these PPPs are extremely complex and poorly understood, so their fate in the environment is still not so known. And although we have a lot of information of the fate and occurrence of these these PPPs in, for example, rivers and creeks. There is still a lot of missing information in other environmental compartments, such as, for example, groundwater, lake systems, and what is the long transport range of these PPPs. Also, we don't have much information of these contaminants in the past because we didn't have the analytical technology existing maybe 20 years ago. So these also have kind of maybe prevent some of the models to predict the fate of these PPPs in the environment. As Sabera mentioned, once that these PPPs enter into the aquatic organisms, they can flow into rivers or creeks, or they can be scavenged. They can absorb into particles and then be in the bottom of, in this case, rivers or sediments. And this is quite concerned because these are the beds of water, drinking water ecosystems, to say it like that. To kind of try to advance a bit the knowledge about this cyclone of the PPPs into the environment, we find a tool in the as let's say I'm the paleolimnology group, so I was interested in sediment cores. And sediment cores are really interesting because there are like libraries, there are like a book of history to pass. And if you can see here a sediment core that goes from the 1920s to now. And also, they can be great sources for changes within a catchment. So then we reconstruct history, and also using the right nuclides to have dating documents, we can reconstruct really history over time. So that will be my metrics. Also, we need analytical methods. We have here we have 80 PPPs, but now we have over 100 PPPs. We do target analysis. However, also we rely heavily on high resolution mass spectrometry and then data analysis of the pre-processing of this mass spectrometry. So here at the right, I'm showing you 3 different cores, and actually these different cores are from the same catchment. So the sampling is can vary between a lake. So it's very important to see where you sample. And on top of that, we need to really understand what are the processes within an aquatic system. In my research, I have acquired or adopted many paleolimnology tools. For example, I use X-ray fluorescence to understand if what we see in the sediment comes from erosion or actually comes from away from the water, or if the sediment has been created within an aquatic system based on carbon and nitrogen ratios, to understand what are the processes being in the lake, also what is the primary productivity, if the lake is eutrophication, has eutrophication or have anoxic waters. And this becomes very important information because a core that is preserved under anoxic conditions can tell us more. So Vera showed in her presentation a sample from Bern, from Switzerland. Here showing a small little map, and this is great because I'm gonna also my talk focus in Bern. And Bern is very important because it's within the Swiss plateau, and it's a place where you is heavily populated with agricultural crops. So here we have a lake that I'm going to talk. We have a golf course in the left side, and surrounded by agricultural fields. It's a small catchment. It's around 0.2 square kilometers, and it's 59% agriculture. So now in my next slide, I'm going to show you some trends over time. And the trends over time that I'm going to show you is really where it has been captured from the sediment water interfaces and is not related to additional processes like, for example, erosion of the soil. Okay, so what we find out is here we have a sediment core, and then we have the H model. And here we have fluxes. We work in fluxes because fluxes take into account the compactation of a sediment. And then what we can find is that we can see or we can reconstruct the deposition of different pesticides since the 1960s. So pesticides have been entered in the environment since the 1960s. And of course, we didn't know because we didn't have the analytical technology. So now, do this this past reconstruction, we can show the different compounds. For example, in this cluster, atrazine that was banned in 2000 and around 2000 it has a trend increasing over time and then decreasing over time. However, it hasn't yet be completely banned from the environment, and that's why we still find it in soils and in water. And we have additional different clusters. Here, some of the clusters start going over time, here as well. And here we have this pesticide chlorpyrifos that was banned in 2029 here in Switzerland. So when we got these results, we were a bit shocked, because if we look at the half-lives of these compounds in sediments, actually the half-lives are within days. And here I'm showing you that these compounds can be extremely persistent all the time if they persist or they can stay under anaerobic conditions. And this is the key. So they are there. So what can the sediment tell us? Using high-resolution mass spectrometry, here I'm showing you a heat map. Each line is a feature, and the more red that you see, the more contamination. We have in the y-axis, the top started in the 1920s, and the right and the left until recent times. And this is, it tells us, when you use high resolution, it can tell us a lot, because we can see that at the beginning until the 1960s, the lake was it was clean. Oops. And then started the era of agriculture around the catchment. But then it started being some environmental policy and water protection initiatives in Switzerland that really helped the lake to kind of be a bit cleaner. However, we start seeing some additional input of different organic contaminants. And when we look at kind of the catchment and development of the catchment, we can see that this is kind of consistent with the building of a golf course in the 2000. So this is how are we using our sediment records to study what is happening in the past, how compounds that are being banned develop in the environment and also around the catchment. So what the lakes can tell us so far, we have different campaigns and different small lakes here in Canton Bern, and we can see that we can find it everywhere, and we can find it since the 1960s. So pesticides have been there. We can even catch these flood peaks. And these are important because a flood peak means like a wash of some, in this case, Difficona sole, and they can cause also for aquatic organisms, could be dangerous. And we found similar pesticides or PPPs in lakes. In this case, we can also approximately really back calculate from the back of the envelope the amount of grams they have been accumulated in the lakes. And if we could put it in a bigger perspective, we find out that the fraction number of individual PPPs in lakes in the Swiss plateau is around 0.1%. And this is consistent with the recent studies of Maggie et al. On the global budget of pesticides, which she concluded this is also the kind of percentage of PPPs that enter into rivers. And this can cause a decrease of species richness of stream and invertebrates, as you can see. So what else can they tell us is if we compare, for example, two different lakes. Mose is the one that I showed you. Now I'm introducing another one, is Bollense. This is a dam lake. This is a bigger catchment area. This is the PPP Diuron. When we go in Mose, we have lower concentrations versus in larger catchments. This is kind of obvious. And the amount and the input in this catchment is going to be greater, right? We have to be careful when we compare also where we sample or when we compare, because our studies are telling us these PPPs get scavenged very interestingly into the sediment. There are like really good scavengers of these compounds, but these samples, these PPPs are not distributed equally into different catchments. So that's why when we go sampling, we always go to the deepest part of the lake, because this is a kind of overview of the whole catchment, but the distribution, this is not equal. Maybe you're thinking, okay, maybe this is only for small catchments, but when we go to a larger scale, like for example, these are also lakes in Switzerland, the north and the south of Switzerland, they have like catchments of 160 kilometers square. We can also see similar trends. The distribution of compounds in certain catchments is very different. And here we have the pesticides. They contribute maybe here wastewater treatment plants, but also around the catchment. So this is kind of important. So now going to maybe talking about the difficulty of comparing sites. We have usually monitoring now around 10 small lakes. We monitor every 10 years water, oxygen, sediment, environmental DNA. This year, environmental DNA. We are comparing to try to see, to make some sense if there are some processes that are in the lake are making the degradation of these pesticides or the persistence of these pesticides meaningful or so on. And where we are trying to, where we find out that is when we compare relative recoveries of a catchment, yeah. Here, it's very important to have exact match internal standards. Otherwise, when we don't have exact match internal standards, the recoveries of different samples from different lakes, it could be vary significantly. And this is very important, because this will shift the matrix factors within your calculation when you, for example, determine the limits of quantification within the matrix. So you can imagine higher matrix factors, lowers limits of quantification. And we start exploring this part because in some of the alpine lakes, we start finding a lot of pesticides, and we were surprised. So when we start doing some recoveries and correcting recoveries of our LOQs with matrix factors, we realized that, okay, most of them, they were below the LOQ. So when we compare sites, we need to be quite careful about the characteristics of the sediment itself. So now I'm telling you, until here, the sediments are present in the lowlands, in the lowland lakes that are close to agricultural areas. That are very persistent under anaerobic conditions, that we can reconstruct the patterns of these pesticides from the 1960s to now. That is not something new. They have been entered already into the environment since the 1960s. But one of the questions is that, if you remember my introduction slide, is that we were also thinking how long this can travel. Or why is we still find them everywhere, and also a lot of these pesticides that have been banned, we still find them. So for that, we have a couple projects going where we sample in very high altitudes. For example, in Oberstockense, this is a lake that is around more than 1,500 meters high up. And we sample even in the winter. You can see here a nice picture of two of my PhD students drilling sediment core from the ice. And what we can find is we can actually find these fungicides which have a atmospheric half-lives of 1 hour of 3 hours. So these kind of PPPs manage to go through really far off from the place of application. Additionally, we can find here, we are investigating also proglacial lakes, like lakes that are forming due to the melting of glaciers, because we are interested about also climate change and how it is affecting the reintroduction and the release of these PPPs and other organic contaminants into the environment. And we can see there are also there, especially some of these fungicides, and these results are consistent with recent publication from Meyer et al., where they say the half-lives of atmospheric half-lives of these many PPPs are really underestimated. So right now I have told you also about they are also on high altitudes, and maybe in what? Maybe the next step that I want to show you is about the toxicity. And because when we analyze a sediment, we use a very hard technique, so we don't know if this is bioavailable or not. And this is an important point of assessing the quality of the sediment. So we have at the moment using environmental quality criteria, and if I can show you to the left, chlorophyphos and diuron, you can see that both of them have been banned. You can see that the risk coefficient sometimes surpass the scale that we have here to the right. So it means that these sediments the quality of these sediments is quite poor. Of course, this is based on calculations. So what we're trying to do is also introduce something called paleotoxicology, and where we use ostracods, and we expose these ostracods to the different layers in the sediment. And we can see, for example, in this case, a high growth inhibition in the top layers. Or after in this case, after 2000, we also see a low or survival, survival of ostracods. So this is something that we also are exploring how this works, because we are interested about how bioavailable are these PPPs in the environment. And also we are trying to assess what is the effect of these PPPs in the microbial community. It's quite difficult because sediment is very complex, but we are trying to use different tools to assess what is the impact of these PPPs. So I hope now I can I can tell you to understand the cycle or PPPs through the environment is really complex. At the moment, we are working with different groups and institutes in Europe to understand how these compounds can be released, for example, from ice, from melting ice, how they can remobilize within lake systems, what is the fluxes between sediment and water interfaces, and how and why these PPPs are traveling really far away and that are really persistent. So I cannot give you an answer to see why this is where we are studying, but I hope I have in this talk I have shown you the sediments are very good sources of information, not only in the past but also in the present, to understand what's going on in a catchment. And with that, maybe I would like to advertise a conference that we host here at the University of Bern every 5 years in contaminated sediments. And this will be hosted here at University of Bern and between the 2nd and 4th of September. And maybe some of you are interested in coming to the University of Bern. And my last slide, I would like to thank you for your attention. And of course, this work is not only my own work, but it's a multidisciplinary work with my group in different master's, bachelor's, and PhD theses. And with that, I'm done. Thank you for listening.
Fantastic, Aurea. That was absolutely brilliant and so fascinating. So at the start of your talk, you alluded to there being some clear differences between the research you've carried out and the research that we've sort of found at Sprint. And I just wondered whether you'd both kind of like to have a brief discussion about that. I mean, are the differences to do with sampling depths and the locations and conditions of samples? Aurea, you mentioned anaerobic conditions, and that might be a key point as well. Or is it differences in terms of the sampling strategy overall and how we analyse the results? It would just be really great to hear your thoughts on that.
Yeah, maybe I just when we analyze spatial analysis, we need to be just careful, because when we grab a 10 centimeter sample from one location, it could be, if you have a sedimentation rate of 1 centimeter per year, you will have 10 years. But maybe you in another catchment, you have 0.3 centimeter per year sedimentation rate, and then you will have completely different results. So when we do spatial analysis, we just need to be very careful when we compare sizes, because the sedimentation rate could be completely different, because it's complex. It's complex.
Absolutely. So it's very context specific. So the findings in one site might be completely different, but that's still, you know, really interesting findings for that particular site. Vera, would you like to elaborate?
Yes, thank you. Yeah, I think this is a very good point, and I'm very happy actually that these presentations came together to also highlight how complex it is to understand and explain these findings. Yes, we do fully agree that indeed the rates, the sedimentation rates are highly variable among our case study sites. At the same time, I think what we also tried to highlight was that despite all of these differences, all these samples from these 8 case study sites, almost all of them have pesticide residues. So that is not a localized problem. Of course, the specifics change, sure, and all these processes and the rates of the place are dependent of the site and the management and the sedimentation and the pesticides applied there. But it's a problem that is like widespread. So the presence of pesticides are there. There are some common patterns in terms of the compounds that are frequently detected. So that is what we were trying to stress, that this should be indeed addressed on the degree level, understanding the processes and the mechanisms, but also keep the big picture in mind, that this is a holistic challenge and that pesticides are indeed like something that needs to be addressed, I would say, more than at a regional level, or even more than at a country EU level. So yeah.
Perfect, thank you. And so we've now got a question from Emily. So Emily has asked, based on the findings in your research, and as the title of the paper alludes to in Vera's case, do the pesticide levels detected pose a significant concern for aquatic systems?
Thank you for this question. Yeah, I was already expecting that question, actually. And to stress that we kept the title as a question mark on purpose. I mean, if it is a reason to concern. So this is a step 1 in the sense of understanding the chemical part of it. So if the contaminants are there, and now we show that they are. And of course, we did some exploratory analysis in terms of comparing the levels of these compounds against toxicological values to understand indeed, like as Aurea also mentioned about these risk quotients. And in some of these locations, indeed, the quality seems to be poor and it seems to be some risk. But these are theoretical exercises. So we do know that especially when you have complex mixtures, as some of our cases, it's possible that compounds interact with each other. So that will be step 2, that is indeed assess the real risk of this. And this is what we are doing also in Sprint. So we have also a work package on the risk assessment. So where we do pick up on the field findings, and we test the mixtures toxicity in a battery of organisms. And we do see that indeed some organisms are more sensitive to these mixtures than others, but I think that are still preliminary, and that hopefully they will come later in this webinar series. But yeah, so I kept it open on purpose, and also to stress that there is also here a bridge between the theoretical risk quotient analysis and the one that once you go to the lab and you do these toxicological batteries, or even this on the paleotoxicological, that is also very interesting approaches to see indeed the risk of these mixtures.
Perfect. And we also have a question from Michael, which is a tricky one because, well, for Sprint at least, we're focused largely on pesticides and plant protection products. But he's asked, how bad is the pollution with pesticides compared against non-pesticide pollutants? So including things like pharmaceuticals, personal care products, yeah, other chemicals, I guess.
Well, that's a great answer, a great question, and I don't have an answer. I mean, we only analyze pesticide residues. Actually, we also analyze copper on this, and we are looking towards analyzing more contaminants in the samples. But I think indeed this is one of the main challenges of fragmented information, so that there are projects and there are publications on pesticides, others on pharmaceuticals, and this integration, this co-occurrence remains a big challenge. So I cannot tell you how they relate to the other levels, because I don't have that information, because we didn't analyze it. And also the comparison, as again, comparison of findings is always tricky because of the analytical and because of the aspects that change also with different studies approaches, so also in the field. So I think it's a crucial question. I'm afraid I myself don't know, don't have data to answer it. But I would love to dig into this.
Maybe I can comment. I think pesticides are kind of the obvious organic contaminant because they are applied on purpose. However, of course, we find pharmaceuticals in certain areas, especially if you have sewer sludge as applied in agricultural areas, you will have it also. We also have a bit of what we call it tourism contamination, where we see in very pristine lakes where they swim, and then we find pharmaceuticals because the people sometimes pee in these lakes. And lately, because of the big commotion of the perfluorinated compounds, we also found highly contaminated sediments with perfluorinated compounds. So this would be also a big issue in sediments, for sure. Brilliant. And if you look at the non-target, and if you look at the non-target compounds, of course, you will have highly brominated, chlorinated, and fluorinated compounds.
Perfect. Thank you both. So I think we have time for one more question. Don't worry if your question wasn't answered. We are circulating documents with all of our answers to all of the questions asked after the webinar. So we haven't ignored you. Your question will be answered if we've missed it today. So we'll finish up with a question from Roger. So he says, it's interesting to see the spatial variation in both large and small water bodies. Do you use any rapid in situ mapping to assess variation to better inform subsequent targeting of samples for full lab analysis?
No, unfortunately not. We could try to do it. We start now to do it in other projects when we look inorganic contaminants like metals, and try to see it, because, okay, it's also a lot of analysis, but now we have a very easy tool that you don't need to do sample preparation, just measure XRF, and then we try to look for a specific hotspots. But in reality, because they distribute quite whenever we see this, something is obvious. You have a wastewater treatment plant and you say, okay, I sample here. You have, but if they travel and they are bound to particles, you also have the air influence, the current. This is something that we are studying, how the currents they can affect the distribution of these contaminants, and also we have seen that the particle size plays a big role, like when they apply to maybe some fraction of organics, or maybe or other fraction of some compounds like to attach to or partition or sorb to certain fractions of the sediment, and this is we are discovering this. It's it's not so straightforward. Exactly.
Excellent, perfect. Okay, so I think we will wrap up for today. So as I said, we will be answering all of your questions. It might just be that we follow up over email. So if you've consented to us contacting you, we will be in touch one more time just to make sure that we've answered all your questions. So the webinar has been recorded, so it will be up on our YouTube channel shortly. Please do follow, subscribe, and give the webinar, well, if you want to rewatch it, feel free, or share among your contacts. We are going to reconvene with this webinar series in January. We're hoping to organise a session about pesticides and the microbiome. So we're going to move into more of our sort of human health findings from Sprint. So we hope that you'll join us then. Please do stay informed on our social media channels and via our website. Fabiana, yes, absolutely. I will email everyone with the question answers and a link to our YouTube channel just to make sure you can all find that. Excellent. Okay, well, thank you so much, everyone. It's been a great webinar and it's been brilliant to see so much engagement, and we'll see you in the new year. Okay, bye.